So my mission today is really two things. One is to tell you some technology derived from C. elegans that might be useful to you. And the second is to give you some interesting biology stories that might entertain you. We'll try both. So our little nematode, C. elegans, is only 1 millimeter in size. And I want you to notice by this micrograph of it that it's a real organism. I've cartooned the different organs. It has an intestine, a gonad. It has all the normal parts of a real organism. And so by studying C. elegans, you actually do study diseases, organ development, cell signaling, all important aspects. In fact, in this room, I teach a course on the human genome. And as I'm preparing my human genome lectures, and looking at the examples I can give from the human genome, I'm always frustrated by the fact that we know more in depth about certain molecules in this little worm and in flies and in the mouse than we do in humans. And so I hope you'll take this seriously. Okay. So in C. elegans, we have used 3 different approaches for genome editing. We adopted through Sangamo the zinc finger approach. Also through Sangamo, we adopted the use of talons. And then with Jennifer Doudna, we adopted the use of CRISPR-Cas9. And I'm going to give you some examples, some early examples from these 2 genome editing techniques, just because it gives you some biology that's easy to describe. And then I'm going to launch into technology for Cas9 and some applications. Okay. So the way we deliver reagents to C. elegans is by taking a little glass needle and taking our genome editing tools and injecting them straight into the gonad. Then we screen the progeny for mutants, either knock-ins or knockouts. So the advantage of C. elegans is that it grows up in 3.5 days. In 7 days, you can have a homozygous mutation. The other advantage is that you can get up to 15 independent mutants from one injected animal. And why do I say that? I say that because when we inject into a gonad, the gonad is an entire syncytium of all of meiosis. In other words, in each gonad orb, there are 2 in the worm, there's 150 separate germ cells, each of which is an independent target for mutagenesis. And so when you bathe these germ cells in nucleases, and repair templates, any single one of these nuclei has the opportunity to be mutagenized, and they're all independent. And so you stick your little glass needle in here, all the materials travel in through here, and finally, since this is a self-fertilizing hermaphrodite, the oocytes come to the spermatheca, they get fertilized, and out pop embryos that will become self-fertile hermaphrodites again. The way we first delivered these tools for genome editing was actually delivering the RNA, which encodes the molecules. We've subsequently gone on to different methods, but these work extremely well. Okay, the other advantage of working with C. elegans is that there's a bunch of sister nematodes. In fact, they go all the way from Pristiacus pacificus, which lives on rotting beetles, to C. elegans, and that covers 300 million years of evolution, which means you can study developmental processes across 300 million years of evolution and learn a lot about the development of molecules and the development of various biological processes. So we have genome editing working in all these species in red. And we do that because the process we study is ubiquitous, and I'll tell you a little bit about it more. So it took a bit to engineer all these techniques across these species. Some are male-female, some are hermaphroditic, they have different kinds of gonads, but it's all working well. Okay, I just want to show you at the outset the kind of mutations that we get from zinc fingers and from talons, and then ultimately from Cas9. There's a motive behind this. So one of our first mutagenic events that we tried was to get zinc fingers to knock out a histone that was tagged with GFP, expressed specifically in the gonads. So you can see these nice germ cell nuclei lighting up with this green histone marker. So we injected our ZFNs, and you can see their target, and we got mutants. And the signatures of the mutagenesis we got were very characteristic for non-homologous end joining. We got abundant deletions, we got insertions and deletions, and the insertions had microhomologies, very characteristic of non-homologous end joining. Same pattern with talons. This is a different gene, different target, same thing. Insertions and deletions, and insertions with microhomologies. I'll tell you a little bit different story with Cas9 in a bit. So these microhomologies are really kind of nice. First, they were a pain, but then they became really useful. Because, for example, we study a process that I'll describe in a while that uses particular binding sites to affect its means of controlling gene regulation. And it turns out that there are these little binding sites. They're short. They're only 12 nucleotides. And as a fortuitous consequence of this ability to make insertions that have microhomology, we've been able to duplicate our little binding sites in a number of places in order to be able to enhance the binding of the molecules we study. So it's been a very handy process that comes from the non-homologous end joining way of happening. Okay. We've also, of course, been able to use these zinc fingers or talons to specifically knock out those motifs that are important. You just guide a little double-strand break right in the middle of one of those motifs, and you can just knock out that motif and not knock out the other motifs that make up the binding site. So you can selectively look at the consequences of eliminating the different motifs and asking what happens to the binding of your protein. So you can very cleverly do an allelic series to figure out how well your protein binds to different targets. And in this case, I'm just showing you how this complex binds. This is a complex, as I said, I'll describe in a minute. For this purpose, you just need to know that if you knock out one of the two binding sites and you assay the ability of the protein to bind to the binding site, you will notice that if you have one binding site missing, you get a much less binding of the complex than if you have the whole thing present. And so you can serially go in and mutagenize individual motifs to actually study the process that you want to study. So that was all fun and entertaining, and the spontaneous mutants were incredibly useful. However, to be a good engineer, one wants to add what one wants to add and take out what one wants to take out. And so to do that, we had to adopt the technology that was actually not so easy in most organisms at the time. And so what we wanted to do was control the insertion or deletion of particular substrates. And the technique we used at first was, in this case it was talons, we used single-stranded oligo templates. Now that's not so surprising these days, but at the time, I think we were probably only the second people to really adopt this approach. These single-stranded oligos are really quite able to serve as targets for homology-directed repair. And so the amazing thing about a worm is that you can inject your little reagents, in this case talons. You can put in all the oligos you want that you want to replace into a particular target. So if you want to make a series of mutations in a gene, if you want to knock out the gene, tag the gene, do anything to the gene, you throw all the repair templates in at the same time. You throw in your genome editing tools, zinc fingers, talons, or Cas9, and you just wait and out pop your mutant. So in a single injection, for example, we were able to convert this HinD3 site, the NCO1 site, into a HinD3 site, an ECO1 site. We were able to get a deletion. We were able to get everything we wanted just by taking all the oligos, putting them in at the same time. So it's a very efficient way of being able to mutagenize the genome. We also were frustrated at the time not being able to make large deletions precisely the way we wanted. And so we were able to knock in these little FRT repeat sites, which are substrates for a flipase. So you knock in the sites. This happens to be one of our binding sites that's about 1 kb. And there's 3 of these little binding sites. We wanted to get rid of all of them, so we wanted to flip out the entire binding site. We put these little FRT recombination sites on either side, and then we just injected RNA that encoded the recombinase, the flipase, and then we flipped out the binding site we were trying to study and ended up with a repaired template that had only this little FRT repeat site in it. So this was early days, and it was extremely useful to be able to finally, in the worm, engineer things. So I've been a geneticist for 30+ years, frustrated by the fact that I had yeast envy. We couldn't change things, we couldn't do things the way we want. So for the first time, when we were able to do this, I felt finally the worm has arrived. We can do anything we want to do, and I was extremely excited. Okay, so again, we extended this across species. Pristyanthus pacificus, which grows on that rotting beetle, was not really very tractable genetically. There was forward genetics, but there was no reverse genetic tools. There wasn't even transgenesis. There was nothing. And so we went in with our little editing tools, knocked in, for example, a flag tagging to this gene that people wanted to study. It's a gene that is important for mobility. We were able to tag it. You can study the properties of the gene. This was unheard of for this organism because finally one had the tools to be able to do what we wanted to do. So these are just approaches that let you know we had derived the means of doing the mutagenesis by zinc fingers and talons. And then with Jennifer's help, we turned to Cas9. And when we first turned to Cas9, it was at the time when we tried single guides, dual guides. The first approach that worked for us was using dual guides. And we went straight back to that histone GFP that was expressed in the germline and knocked out the histone GFP. So that was really nice. And so we were extremely excited to be able to use Cas9. But then we started running into trouble. So we ran into trouble because we could never predict what a good target would be. We could never predict what a good guide would be. And our success was not wonderful, and we were very confused. So we set about, Bobby Farbood in my lab, set about to try and figure out different rules that might help the efficiency of Cas9-mediated mutagenesis. And so what I'm going to show you is our early problems. So in the literature, there was a guide. This was by the Clarka lab. This particular guide to a gene called CLIP12 worked extremely well in their hands and in our hands. If we just injected the Cas9 with its little guide, 52% of the animals that that received the Cas9 molecule were mutagenized. That was great. We thought this is going to be a breeze. But then we tried 3 other targets. We're good injectors, we're good geneticists, 0. Actually, 4 other targets, 0. We couldn't predict the guides. We couldn't predict what a substrate was. So Bobby, having listened to a talk from Jennifer's Doudna's lab, had a really good idea. Jennifer's lab had shown that Cas9 likes to bind to NGGs. It will happily go to any NGG. And this led to the idea that if one had NGGs around, one might increase the frequency with which Cas9 would find its target. But Bobby got a little more sophisticated in thinking about that. He thought, what if we try a very specific kind of approach and ask the question, does it matter where the NGG is? So he did the following clever set of experiments. He made 2 kinds of guides. He made one guide in which the PAM, the GGG, was here, but then the 3 nucleotides, the 2 nucleotides at the end of the guide-specific, the target-specific sequences, he made GG. So by this approach, you have NGG, NGG, a higher concentration of G. And he asked whether that would be successful. But since he's a good scientist, he did the control experiment as well. He made another guide that was shifted by only 3 nucleotides, 3', such that that NGG, which was formerly part of the guide, now became the PAM. And so the only difference is whether the NGG is at the end of the guide or whether it's actually in the DNA adjacent to the PAM. And so he asked, which one of these is successful, if any? And his result was really quite striking. So the way he did the experiment was he injected Cas9 in the guides. He also injected a transformation marker that lets you know which animals actually received your genome editing tools. And then he assayed, it happened to be a red marker that made the body wall bright red. He then assayed, picked those worms, let them lay progeny so he could collect his mutants, but went back after the mother laid progeny, ground her up, and assayed her to see if she had a mutation. And then he collected the mutations. And it was stunningly successful. So the first guide he made, these were the 3 genes that he was not successful mutagenizing previously at all. He stuck a GG at the 3' end of the guide, and it went from 0% to 72%. He then used the shifted guide such that the GG was no longer part of the guide, but instead adjacent to the PAM. It became the PAM. The frequency went down to 0. He did this several more times, same story. Very high frequency of mutagenesis with the GG, you shift it by 3 nucleotides, 0. And so what this meant was that there was actually a way of predicting, at least for C. elegans, what targets and what guides might be successful. And so far, we've done 25 guides this way, and all 25 have been successful. So at least for the worm, this has been highly useful. Okay, So our median mutagenesis rate is 51%. The range is 10% to 72%. With the 3' GG shifted guide, the median mutagenesis rate is 1%. So at least that meant that we knew what we should target, and we could do it efficiently. Okay, so now I want to talk to you briefly about what kind of products do we get when we do Cas9-mediated mutagenesis. We get the standard suite of things in that we get the majority of things we find are deletions. These are all non-templated, so this is just what happens if you put in the guide and the Cas9. We get abundant deletions, we get insertions and deletions, and we get insertions. But the interesting question is, what are these insertions? And this is the strange part that Dana and I are still discussing. I'm not sure what it means exactly. When we get insertions, our insertions do not look like the characteristic ones with huge microhomology. They look like little additions. And so this raises the question of what molecules in the worm are being used to actually repair the double-strand break, and is this really non-homologous end joining by the same route that's used in other organisms? Are there other enzymes in addition in C. elegans that are actually doing this repair process? And we're investigating this. Okay, so just to kind of give you a feel for whether how general this process might be. Of course, you're very well familiar with the fact that many important studies have been done in mammals and zebrafish to ask what's important about the guide. And there was several reports that the terminal G is really important, and there are other reports that said that really just purines were important, and that's clearly true. But what I want to show you, both from our data and other published data from C. elegans, is this is not sufficient to guarantee that your guide will work. And so if we just look at our own data set, I showed you before that all these GG guides worked very well. But if you actually look at guides that have a terminal G, they give you varying success. In other words, a terminal G does not guarantee that this guide will find its target and work well. Simply having purines doesn't guarantee anything for the worm either. And then, of course, T's and C's aren't so favorable either. And so this says that, at least for C. elegans, there's something magical about these GG guides that's important. It's possible that the mutagenesis rate in the worms is lower than in other organisms, and it's possible that this then elevates it to a particular level. It's also possible that in other organisms the rate's so high that it matters much less what your guide actually is. Okay, so if you look at all the literature on C. elegans, the guides that were the most successful were GG guides. The non-GG guides in C. elegans, if you just look in the literature, the median was 4% mutagenesis, not 70%, not 50%. So for at least C. elegans, it's important. It may be important for other organisms, but it's something you should keep in mind. Okay, so then having been able to convert the use of Cas9 into something we could predict and felt really good about, we asked whether we could then use homology-directed repair templates to insert or delete whatever we wanted precisely. And that turned out with these guides to work equally well. And so you're very familiar with this. We pick a target. We pick a repair template. It can either be a single-stranded oligo, or in this case, it's a double-stranded piece of DNA with 500 base pair homology arms. When we design a template for repair, we mutagenize the PAM in the repair template so we don't get continuous mutagenesis after we insert what we want. And so you just inject your repair template, your Cas9 in the guide, and you hope for this. And what was striking was that it worked incredibly well. So in the case of this example I'm showing you, without the double-stranded repair template, our frequency of mutagenesis was 72%. When we put in the HDR template, everything got converted to actually a homologous recombination. And so the frequency, basically every product we came out was repaired correctly by HDR. And so that said that you had your choice. You could either knock something out or knock something in. And so that was very exciting to us. Okay, but we were not entirely satisfied. We don't like to work hard. We were not entirely satisfied that this was the most efficient way to do mutagenesis. And Andy Fire had come up with a really brilliant approach. that we adopted in combination with our GG guides, which turned out to be even more efficient. Okay, his approach was this. He called it co-CRISPR or co-conversion. And the idea is you inject 2 different guides to 2 different genes with your Cas9, and you ask for the guide that is your control guide to go into a gene that when you insert a template gives you a dominant phenotype. In this case, it was a roller phenotype. And what you do is you take those worms that just are visibly rolling and ask, are those worms that are rolling also mutagenized in your gene of interest? If that's true, you do 1 injection, you let the animals lay progeny, you pick only the animals that are rolling, and then you assay only those. And that was Andy Feyer's invention, and it worked brilliantly. Okay, so this is the approach. You take your worm, you inject a guide to roll 6, and a repair template that causes a dominant gain-of-function phenotype when it gets inserted. You also inject the guide for your gene of choice and the repair template for your gene of choice. So you inject all this stuff into the worm. You only pick the rollers. Since it's a dominant gain-of-function phenotype, you know that you should assay this mom, but you let her lay progeny. And you don't really want that roller phenotype in your ultimate worm. So what you do is you let that roller mutation, which is gain of function, segregate away, and you pick the non-roller animals from the roller animals that actually not only had mutagenized roll 6, but also your gene of choice. In other words, you're guided to what genes to look at what animals to look at by PCR, by the roller phenotype. And once you know that the roller animal had what you wanted, you let that gain-of-function phenotype segregate away, and you just pick your animal that has the correct property. This worked astonishingly well as well again. And so in this particular case, we injected roll 6. We injected a particular gene. We actually had tried, I can't even tell you how many times, to get a particular insertion into this gene. It had always failed. But with using, just because it turns out to cause some problems, but in this approach, if we took the rollers and we assayed the rollers for the presence of the mutation we wanted in our gene of choice, 63% of the rollers also had the correct insertion that we were looking for. 16% had non-homologous end joining, and 21% were wild type. So again, we reduced our how many animals we had to screen dramatically by only taking the rollers, and we got easily what we wanted. And in fact, this is the better strategy. So we have converted completely to this idea where you inject 2 markers, you look for something that has a mutagenic event that's easy to spot, and then you interrogate the gene of interest for yourself. Okay, so in worms, sometimes we want to make bigger deletions. In this case, we wanted to knock out a gene that was 3 kb. We decided to use the approach of having 2 guides to make 2 double-strand breaks to really mutagenize this site, and then bring in a repair template to correct this error, and bring in a repair template that included, to knock out the gene and included a stop. And part of the reason I'm showing you this is that this frequency of mutagenesis is so good that when we put in 2 guides plus the roll 6 guide, 33% of the time we got complete deletion of the gene and homology-directed repair insertion by using these 2 double-stranded breaks. So when you want a big deletion, it's best to flank it with double-strand breaks to repair in what you want. There's actually another reason I'm showing you this example. And the reason I'm showing you this example is sometimes you get what you don't expect. And this is just a cautionary tale. So as I mentioned, we use this roll 6 oligo to repair in a gain-of-function phenotype into roll 6. We have our gene of choice, and we knock in or knock out whatever we want, and we bring in a homology-directed repair template for that. But what was kind of shocking to us was that we would get unexpected mutations. So we would find that the oligo that we wanted inserted in our gene X actually got inserted at roll 6. It didn't cause a roller phenotype. We always interrogate roll 6 regardless. But the point is that if you have single-stranded pieces of DNA floating around in your genome they're going to be inserted when there's double-strand breaks. In this case, it's pretty dramatic because you intentionally put a double-strand break at high frequency in one place. But these are completely illegitimate recombination events. These are non-homology-directed insertion of this single-stranded template. It's highly mutagenic. And I only tell you this as a cautionary note. When you're busy mutagenizing stem cells and human cells, it's really important that you think carefully about the design of your experiment. And it's really important that you know you can get rogue events at frequencies that are pretty high. So this is not just off target. This is insertion of an oligo in a place you don't want. And so you actually need to think about using sequences designed to that, whatever that oligo brought in, to see if it's elsewhere in the genome, because it might be. Okay. So from that, we concluded that these 3' GG dinucleotides were really, really predictive of high mutagenesis rates. And so that was very exciting to us. And that the frequencies are great, and they're great for either non-homologous end joining or homology-directed repair. And that coupling this 3' GG guide with this co-conversion strategy really enhanced the mutagenesis frequency and they simplified the screening. So for us, that was really important. There have been some other clever technologies that I want to describe for you that might be useful for you. And this particular one has come from Bob Goldstein's lab. The first author of the paper is Dickinson. And this is a particular way of genome editing that might be useful for cases where the frequency of mutagenesis of the target you want might be quite low, and you might want a selection. And so this is a really clever approach in which you actually insert a self-excising cassette, which when you excise it, gives you the product you want. So let me describe the cassette and let me show you an application of this. So this is a cassette in which there's fluorescence protein, in this case green, and it's flanked by a LOX-P site, and there's 500 nucleotides or so of homology on either side. The cassette has Cre, and it has a hygromycin resistance gene in it. And so Cre is a molecule that can excise whatever is between the LOX-P sites. And hygromycin resistance is something you can select for. So if you use Cas9 to target this cassette into a particular locus that you're interested in, then all you have to do is select for hygromycin resistance. That guarantees that this whole cassette will have gotten in. And so you can select on cells, you can select on animals for hygromycin resistance. Okay? And so that makes your job easy. You slap things in the incubator, you go away, come back and look at what's alive. Very easy. But then the clever part, in addition, is that since this Cre molecule is under the control of a heat shock promoter, you can take those cells or those animals that have the cassette you're interested in inserted, you heat shock, that makes the Cre, which then excises the cassette through the LOX-P sites. And when it excises the cassette, what you end up with is your gene that's now tagged with a fluorescence protein and another tag. And the little LOX-P site is simply in an intron where it usually doesn't matter. And so that lets you select what you want, tag your protein. In this case, the application I'm going to show you is a little bit small, but it's the same cassette. It just says SEC here. In this case, even before they've heat shocked, the way they've engineered this is that the GFP is actually butted up against the promoter of the gene they're interested in. This happens to be a histone promoter. And so even before they excise the cassette, when they just have hygromycin resistance, they can actually interrogate the transcription level of the promoter of the gene they're interested in. So this enables them to make a transcriptional reporter without further work. And then if they use heat shock to excise the cassette, they then have a protein that is tagged. And I'll show you what that looks like. So this is a little worm before the cassette has been excised. So this is just a transcriptional tag off this histone promoter. And this is once the hygromycin, the cassette has been flipped out. you get only the nuclei staining because it's a histone marker. So this is an extremely cute technique. There's no reason it can't be used for cell lines or for anything else. It's efficient, and it gives you a lot of tools. Okay, so I'm now going to turn to some biological examples to show you how editing across nematode species has actually been useful. Just a couple lessons of biology. And I'm choosing one of my favorite topics, the topic of dosage compensation. So in humans and flies and worms, sex is determined by some sort of chromosome mechanism. In the case of worms, hermaphrodites have 2 X chromosomes, males have 1 X chromosome. The animal can count the difference between 1 and 2. In humans, as you know, the Y chromosome determines sex, it determines maleness. In flies, it's a similar mechanism. And so organisms that have this kind of sex determination mechanism, counting chromosomes or using chromosomes, have a problem. The problem is that the 2 species have different numbers of sex chromosomes and therefore would make different levels of products off of these chromosomes, but often the products one needs in a male and a hermaphrodite are of the same level. And so the poor male would be at a disadvantage, or the poor hermaphrodite would be at a disadvantage by having the wrong level of gene expression. And so in flies and in humans, mouse, rabbits, nematodes, mechanisms have evolved to equalize gene expression, and those mechanisms are called dosage compensation. And we've been able to find a large complex called the dosage compensation complex that in this case binds specifically to both hermaphrodite X chromosomes and reduces transcription by half so that the level of transcription from these 2 X's equals that from a male. And the complex of proteins that does this is actually a complex that was stolen from all of biology. It's a condensin complex. So the condensin complex, first described by Doug Koshelin and Tatsuya Hirano, is a complex that's important for compacting and resolving chromosomes in mitosis and meiosis. And the worm stole the complex it had for those purposes, and it modified it and created other proteins so that that complex could be targeted specifically to the X chromosome. And the implication of that was, many years ago when we first discovered this, was maybe this dosage compensation complex folds up the X chromosome in some way that makes transcriptional repression easy. And it turns down transcription by changing the conformation of the X chromosome. I should add that the worm was really clever in what it stole for its dosage compensation complex. Not only did it steal condensin, it stole a protein from a completely different kind of complex, a complex that's involved in activating gene expression, the compass MLL complex. This complex is involved in putting the trimethyl mark on H3K4, which is a hallmark of actively transcribed genes. And so when this protein is part of this complex, it's a gene activator. When this protein is part of this complex, it's a gene repressor. Okay, so you all know that chromosomes are not simply randomly oriented in the nucleus. They actually are quite ordered, and chromosomes actually come in territories. And so each of these little colors represents the color of a chromosome. And they're not random. They're quite organized. And not only are they organized in that chromosomes tend to be bunched together, they're organized with regard to what part of the chromosome is attached to the nuclear periphery and when. And attachment to the nuclear periphery and attachment in various locations turns out to be extremely important. And so one question one can ask by studying dosage compensation is, what is the role of gene expression? What is the role in gene expression of chromosome structure? And because our complex was a condensin complex, we could study this. 2 more facts. When this complex binds to the X chromosomes, we discovered it binds through specific binding sites. Those were those binding sites I showed you before that we deleted. And there's these 2 motifs of these 2 different sequences, which are really enriched on the X chromosome and really important for bringing in the dosage compensation complex. So you can imagine what you can do with Cas9. Okay, so what I'm going to show you is that the condensin complex actually folds up the X chromosome in a different shape in hermaphrodites. And I'm going to cartoon this for you. I'll show you a couple of heat maps to show you I'm not lying to you. But this is the X chromosome, and it gets organized into specific domains. These domains are about 1 megabase each. And the way it gets organized into domains is when this complex binds to its recruitment sites, it actually creates those domains, pretty shockingly. And just to show you I'm not lying, the way we approach this is an approach called Hi-C in collaboration with Job Decker. Hi-C is chromosome conformation capture done genome-wide. I'm just going to introduce what it is. If you take your worm, you take any organism, and you fix the DNA, you're going to trap any conformation the DNA is in. And DNAs that are adjacent to each other tend to interact with each other. You can get loops. And if you're trying to interrogate the structure of an entire chromosome, you're interested in what part of the chromosome is talking to what other part of the chromosome. If it gets folded up, are they actually interacting? And this approach of Hi-C will capture those events. And so Hi-C, you fix your DNA, you digest it with something that will get rid of those loops because it's a frequently cutting enzyme. You religate what's left, and so you then release the ligation and you purify these little molecules and you sequence them. So by sequencing these short molecules that remain, you're able to find out what piece of the X chromosome or any chromosome is attached to what other piece. And I'm just going to show you what a heat map looks like, and then I'm going to go back to cartoons. So this is just to show you that I'm honest. So you display all the properties of that genome-wide sequencing in a heat map, and each of these represents the location on the X chromosome in the X-axis and the Y-axis. And this is a diagonal. Any piece of DNA that's adjacent to itself will interact with itself, and you'll trap it by cross-linking. That's not interesting, and you expect to find it. What's interesting is if there are inner outliers. You see these little boxes? You see how organized this little X chromosome looks? You see all these self-interacting domains. So I had cartooned them for you looking like this. But in real life, they look like this. You see these boxes. And that means pieces of DNA within this genomic location are interacting with each other. And then there's a boundary where they don't interact with each other. And then there's another self-interacting domain. These are called TADs. And what I want you to notice is that the X chromosome looks very different from a non-X chromosome. The non-X chromosome has very little structure to it. The X chromosome is highly ordered. And so now I'm going to show you, this is that little domain, and that's that boundary. And I'm going to show you, just again by heat map, that if we eliminate the dosage compensation complex, the condensing complex, those topologically associating domains go away. And so here are those domains again. I've just switched the orientation. These are the boundaries we call by genomics. We mutate the complex, and what I want you to see is really those boundaries go away. You start to get interactions across the entire chromosome. And we've shown by a lot of experiments that these boundaries are at those recruitment sites, those rec sites. So the obvious experiment to do is to use Cas9, knock out one of those binding sites, and ask if the structure of the X chromosome in that local region is abrogated. And so we took Cas9, we knocked in an NCON site, and we knocked out one of the recruitment sites that are actually at one of those boundaries. And we interrogated what happened, and this is the result. We were able to knock out one of the rec sites that was actually mediating the boundary, and the boundary completely went away, and the structure was abrogated in that region. Okay, so that's one application that was extremely important for Cas9. It lets you really interrogate very precise things and ask biological questions. It really is the dosage compensation complex that creates a structure. It really creates a structure by binding to its binding site. Knocking out one binding site can really change the structure of a part of the X chromosome. So that was really important. And so obviously what we're doing is we're deleting all the rec sites at the boundaries to see what happens. We're inserting rec sites in different places on X and on autosomes to see if we can create the structure. And so that's now made very easy by Cas9 technology. I'm going to give you one more example, and that is the evolution of dosage compensation across nematode species. So the fact that you have a large protein complex that's conserved, it's condensin, makes you wonder, is this the complex that's used in other organisms, in other related species? And if so, what does it bind to on the X chromosome? Is it the same sequence? Is it a different sequence? Is part of the evolution of species changing the X chromosome so that you have incompatibilities between species? So we looked at these 4 Cenobditis species. They diverged by 30 million years, to ask very simply using Cas9 to figure out whether the protein molecules are the same. Is the dosage compensation complex the same? And secondly, what does it bind to? And this is very brief. It's a very nice story. So we asked whether the condensin complex was conserved, and we asked whether the loaders of the condensin complex were conserved in these 4 species. And so we went in with Cas9 and we tagged the relevant proteins in those species. We stained with antibodies against the tag, and we discovered that in those species, both a condensin subunit and a loader was found localized happily to the X chromosome in all of those species. It said that across 30 million years of evolution, it was using the same proteins. We knocked out the proteins. They disrupted dosage compensation. So that was all cool. And so the complex is definitely conserved. But what about the binding site? Okay, so we wanted to know what motifs confer binding of that condensin complex in these different species. So since we had conveniently tagged the molecules that are part of the complex with flag, we could then go in and use that tag to do ChIP-seq experiments to find out what the binding sites were, and then find out what motifs were important. And this is pretty interesting. We discovered first, when we looked in the C. elegans versus this group here, C. Briggsii and C. negoni, we discovered that there was a motif similar to the C. elegans MEX motif. However, it was diverged sufficiently that when we put these motifs back in C. elegans, they didn't function. And when we put C. elegans motifs back into these species, they didn't function. So they look somewhat similar, but they're not functionally related at all. However, C. negoni and C. Briggsii are so close, and the motifs are so close, that in fact they do substitute for each other. They're slightly different, but they do substitute. Okay, so that said that the first motif was similar between these 2 species, but very different from C. elegans. C. elegans also had another large motif, and we found 2 large motifs in C. negoni and C. Briggsii. They look very similar to each other. They substitute for each other, but they completely look different from C. elegans. And C. elegans doesn't substitute for these, and these don't substitute for C. elegans. So clearly, across 30 million years, there has been a divergence of the binding site. But the question becomes, when did this divergence happen? How rapidly evolving is the X chromosome? Because most protein complexes that are conserved, that you know about, that are involved in body formation, have a binding site that's conserved from Drosophila to man. And so it's highly irregular that a very conserved protein complex can change 100 binding sites in a very short period of time. And so we wondered, how rapidly was it changing its binding sites? And so since we knew that these 2 species were very similar, but yet quite distant from C. elegans, we wanted to know when it diverged. And so to do that, you look at an outgroup, in this case, C. tropicalis. And there are certain specific predictions. Was the divergence here? Was the divergence here? Was it in both places? And I'll show you the predictions. So if tropicalis is similar to these 2 guys, then the divergence occurred here, when C. elegans split off from tropicalis. If tropicalis is similar to C. elegans, then the divergence occurred here. If tropicalis is different from both these 2 and from C. elegans, it diverged twice. And this turns out to be the answer. It's really rapidly evolving. And so we were able to do this, again, by tagging all of our proteins, by knocking out our proteins, by doing ChIP-seq experiments, by finding motifs, by knocking out motifs, and asking what's important. And so the conclusion there is that the dosage compensation complex, the components are functionally conserved. but the binding sites have remarkably diverged. They resemble each other somewhat, but they're so different that they're not functional across species. And there must be some sort of coevolution of the dosage compensation genes in order to change their binding specificity, or we're missing something like a non-coding RNA that's involved in changing specificity. We don't know. But this is a heavy level of investigation, and it's greatly aided by all these genome editing tools that we've devised to go across species. So with that, I'll stop, ask for questions, and hope I've at least entertained you with some thoughts about what to do in some interesting biology. Thanks.